the role of analytics in key collators within this model is effective and only revealed in female animals. So I'm going to talk about the conclusion. So I think many of these statements were already brought up yesterday, especially in that second text talk. But just to go over our particular findings, analytics, first of all, we showed it does not have a significant impact on intestinal microbiota homeostasis. And we say, of course, that the use of limiting mice is really the gold standard for normalizing not only genetics but also environmental variability in animal models. The sexual microbiome is impacted by sex. Sex hormones are no influence of the microbiome, and this was previously published by Mycology and Possible for Children in Toronto. And finally, analytic in female mice, but not males, has a decreased severity of GSS in these colitis compared to wild-type females. So there seems to be an interplay of analytic genotype and sex, but independent of the microbiome, that increases colitis severity. So getting to the critical challenges in understanding the role of the microbiome in the model, I think this study really demonstrates that the use of litter mice from heterozygous breeding is imperative for well-controlled studies. I think the thing that we need to think about is that the microbiome influences on phenotypes are not just restricted to gut. There's a lot of studies showing that the gut microbiome can influence other models in animals independent of gut. So how can we better control these studies? There's a lot of studies out there that co-house mice. Is it sufficient to normalize the microbiome? Our data says it's not. You really need to use litter mice from mothers or related mothers to have a well-controlled study. And the other challenge we have is how to control microbiome effects in complex backgrounds. So many people, for example, they have double knockout mice, so people triple knockout mice. How do you litter control for this kind of complex genotypes in mice? Lab reproducibility is a huge issue, so multi-center validation, so we contact colleagues and see if they can validate our findings in other facilities. Will we need to move to a standardization of the microbiome using defined communities? And also, as we recently showed, there's certainly other microbes that impact phenotypes. We have paper recently showing that the parasites have a huge impact on the gut at baseline. So parasites, for the other organisms also should be considered. Improving this, we need to report source, so this is also taken from the recent review from FAB and the state version. We need to really report source and sex, so genetic backgrounds, control the microbiome, discuss the breeding strategy that we use to generate litter mice, the animal numbers as well as we showed in our study, important to also include sex within this, and also the cooperation among stakeholders, funding institutions, editors, reviews need to reinforce these reporting rules. So in there, sorry, I'm going to go back. I would thank the people in my lab that did this work, so particularly Stephen Robertson, who's my microbiome expert, and Paul Lemire in my partner's lab, Patricia Farzan, with help from David Gutman and Adam on the field of microbiota bioinformatics analysis. So thank you very much.
So our next speaker is Dr. Nita Trumpman from Medical College of Wisconsin, and she will present antimicrobial peptides shape intestinal bacterial niche creation and competition.
Thank you for the invitation to speak to you today. I think we'll get a little bit more from the side of the microbe and the host. The study of my lab has been antimicrobial peptides and factors that shape the intestinal microbial niche. We know and have been reported in the last day, and we'll continue to discuss that there are a number of factors that shape the intestinal microbial niche, and the one I'm going to be speaking about today is antimicrobial peptides, which are immune effectors. Antimicrobial peptides are an ancient and conserved mechanism of host defense. We find them from plants to bacteria to fruit flies, moths, and humans. And we know that they have a role in fighting off pathogens, but we also are looking into the role of regulating the commensal microbiota. So there are a number of sources of intestinal antimicrobials in the GI tract, which is the area of my interest. We know that tanner cells, and this is a study from my lab and others, produce a wide variety of antimicrobial peptides, including antimicrobials. The liver produces bile acids, which are antimicrobial, and then microbes actually are a very rich source of antibiotics in the gut through the production of metabolic byproducts, bacteria, enterocytes, and other toxins. Work from my lab a number of years ago looked at the role of enteric defensins in the regulation of the intestinal microbiota, and this is looking at defensins in tanner cells. This is an EM from Andy Wallet's lab of a tanner cell, which is down at the base of the small intestinal crypt, and cells line mucosal surfaces in the small intestine at the base of the crypt. They produce and secrete a wide array of immune effectors and antimicrobials, including defensins. And work from our group showed that if you change the composition of antimicrobial peptides within the tanner cell, this is using littermate controls with either reciprocal models of adding a human defensin or removing production of any tanner cell defensins, we see reciprocal shifts in the microbiome. And then we also found that if you alter the composition of tanner cell antimicrobial peptides, you alter the composition of the intestinal microbiota, and we and many other groups, of course, have shown that if you alter the microbiota, you alter mucosal immune responses, and obviously you alter a lot of the animal physiology. So then we turn to another question, which was, what is the role of antimicrobials produced by commensal bacteria in either forming or competing for an intestinal niche? And for this, we turn to use Enterococcus faecalis, which is a gram-positive coccus. It's a facultative anaerobe, and is a commensal of both animals and the human GI tract, and it's actually quite broadly bound as a commensal. It's intrinsically resistant to cephalosporin antibiotics, and it's also extremely good at picking up antibiotic resistance genes via Barthel gene transfer. This has resulted in the development of multi-drug resistant strains, which are common causes of hospital-acquired infections, both ETHCM and ETHCalis, and they've been designated serious public health threats by the CDC. So our interest was to investigate how antimicrobial peptides contribute to Enterococcus faecalis niche formation and competition in the GI tract. To do this, we needed to generate an animal model that did not disrupt the GI tract with antibiotics, because if you disrupt the GI tract with antibiotics, you're no longer studying colonization, you're studying infection. We did this by feeding mice with Enterococcus in their drinking water for 2 weeks, and after withdrawing Enterococcus from the drinking water, we followed enterococcal colonization in the feces and then apparatus GI tract portions. And we found, this is showing over 4 weeks, but we've done this as far as 11 or 12 weeks, which is as far as we've taken an experiment, and we find continuous shedding of Enterococcus from the GI tract of these mice. And when we sacrifice the mice, we find that there's consistent, stable colonization of the mouse GI tract without prior antibiotic disruption. So now we're able to get a lab strain of Enterococcus to colonize the mice, and this allows us to look at how it forms its niche and how it competes for its niche. And it turns out that Enterococci can harbor plasmids, pheromone-responsive conjugated plasmids that produce bacteriocytes. the plasmid of our interest is the one depicted here, which is PPD1, and it harbors bacteriocin 21. And what we find is that bacteriocin, which are antimicrobial peptides, they can specifically target and kill bacteria of the same genus, and they act by damaging cell membranes. And this is an example of AS48, which is essentially the same as bacteriocin 21. It disrupts cell membranes, and it is broadly anti-gram positive in vitro. Even though these are considered to be somewhat specific, this is actually a relatively promiscuous bacteriocin. So the bacterial strain that harbors a bacteriocin plasmid also encodes an immunity peptide, and that is used to protect itself from being killed by its own bacteriocin. Enterococcus produce bacteriocin to give them a competitive advantage, and alternatively, under the correct circumstances, instead of killing an other organism with its bacteriocin, it actually encodes also what's called a conjugation apparatus, so that it actually directly can contact another enterococcus and transmit that bacteriocin to an enterococcus that lacks it. And this requires direct cell-cell contact, and we actually use that back to our advantage to understand the niche in the GI tract. So what we found in this experiment, where we were just looking at culture of enterococci in the GI tract from mice that did not receive enterococcus, mice that got our enterococcus strain that lacked the bacteriocin, and mice that harbored the bacteriocin, the mice came with a full complement of enterococci when they arrived from the vendor. If we colonized them with lab enterococci without bacteriocin, they colonized at a low level, but persistently. If we colonize with enterococci that had the bacteriocin, it colonizes at a much higher level, and essentially overtakes the entire niche of enterococci in the GI tract. If we knock out the bacteriocin, part of the bacteriocin operon, here we have enterococci colonizing, here we have the bacteriocin enterococci colonizing, and here we have the knockout. It no longer can colonize. It turns out if we restore the bacteriocin operon on a separate plasmid, it regains the ability to colonize, but it does not regain the ability to conjugate, and we use that also later. So the other thing we noticed is we're looking at what happens, whether conjugation or competition is what's going on in the GI tract. So we first colonized mice with enterococci that lacks bacteriocin, and then chased in what we would call our regular strain that produces bacteriocin. And what we found, this is looking at transconjugates, this is looking at the first organisms and which ones have picked up the bacteriocin plasmid. And over the period of 5 weeks, 100% of these strains that are remaining have actually picked up bacteriocin. So this suggests that when delivered sequentially, the enterococcus harboring a bacteriocin can overtake the entire niche in the GI tract. And this also tells us something very important, which is that enterococci localize to a specific physical niche in the GI tract and directly physically interact with other enterococci in the GI tract. We thought we could employ this therapeutically, and so we did. We looked at VRE, and up here we colonized mice fine with multi-drug resistant enterococci. In this case, we used an enterococcus, our complemented strain, because we did not want to conjugate bacteriocin activity to a multi-drug resistant strain. We thought that was a bad idea to get another mechanism to colonize. So we chased with a strain that could produce bacteriocin, but couldn't conjugate it. And what we see is over several weeks after training the mice, we can almost completely eliminate colonization by VRE with the administration of this bacteriocin-producing strain. We find that because we use a complemented strain, we did not conjugate plasmids to the VRE strain. What was also interesting is we looked at the microbiome, and we saw that when we compared the microbiota of enterococcus colonized mice versus PPD1 colonized mice, there were some significant but subtle differences in the microbiome. They were entirely driven by changes in mucospirillum shaleri. It turns out that mucospirillum is not gram-positive and is certainly not enterococcus. So we believe that these changes are indirect changes and not directly caused by direct killing from the bacteriocin. And we just compared it to conventional antibiotics, such as mice that are treated with streptomycin, and we see much more significant differences in colonization between mice treated with streptomycin, say compared to mice given bacteriocin-harbored enterococcus. So unlike conventional antibiotics, bacteriocin 21 primarily impacts enterococcal populations, and this suggests that we can actually therapeutically target a specific niche in the GI tract by engineering organisms. This takes advantage of two aspects. One is it takes advantage of the native biology of the enterococcus to gravitate to the right place in the GI tract, and then it takes advantage of the specificity of the bacteriocin to specifically eliminate the multi-drug resistant strain. It turns out this strain works on ECCM as well. But then we found a little hitch in what we were doing, and this leads to my discussion of the knowledge gaps that need to be addressed before we can advance these types of treatments. We looked at gene expression from these mice, from the guts of these mice that were treated either with nothing, or with enterococcus alone, or with enterococcus that harbored bacteriocin. We isolated RNAs on each of these segments of the GI tract, and then we see that the control mice are clustered very closely together. The ones that have just been colonized by our lab-treated enterococcus actually cluster quite separately from our controls. And those that receive the enterococcus harboring PPD1 cluster separately as well. So this suggests that we have differential gene expression driven by the colonization and also by the PPD1, the effect of PPD1. And this is actually a major knowledge gap if we want to therapeutically intervene in changing the microbiome. This is an important translational goal, intervention, but it may lead to unintended consequences. And I apologize for the stick drawing and all the arrows everywhere, but essentially we don't have the slightest idea at this point what is affecting what in the GI tract. You know, these microbes we know are producing this toxin, it's killing other microbes. Are we signaling the host through the gut as microbial peptides? Is this organism directly signaling the host? Are other metabolites signaling the host? Are we directly changing other members of the microbiota, or is the host response changing other members of the microbiota? So we know that we have unintended consequences here, and we need the knowledge for precise intervention. So our knowledge gaps, what I consider a major knowledge gap is a better understanding of microbe and micro-host interaction in the GI tract, which will allow more precise manipulation of populations. This will entail the development of good systems, genetic systems for genetic manipulation of commensal species, a greater understanding of the bacterial physiology of commensal species, pneumobiologic systems with graduated complexity to take apart these interactions, and also the identification and characterization of physical and metabolic subniches within the gut. And many of these things have been raised over time through many of the speakers over the last day. The collateral benefits to this, other than precise intervention, is just having a better understanding will enhance our ability to make sense of our large data sets in our current microbiome studies. And also, it should allow more appropriate sequence annotation and improve our database accuracy, which is clearly a concern for understanding our current studies. And finally, the people who did this work are here. Most of the work I talked about today was done by Sushma Kamani, who's a very gifted postdoc in the laboratory, a large group of collaborators. Chris Kirstich is my collaborator for all of our enterococcal genetics, and our funding agencies, of course, and somebody about ready to check it out. Thank you.
great presentation and 30 seconds less than the 15 minutes. So we have our last speaker for this session, Dr. Ramling Javier from Harvard University and co-president of Microbes, Molecules and Mucosal Immunity.
So I want to thank Lisa and the organizing committee for this invitation. And what I'd like to do in the next 15 minutes is to give you some highlights of how we're approaching the gut microbiome in health and disease. So broadly, the gut is primarily involved to absorb nutrients, and despite sort of years of understanding of this process, I think there's still a deficiency in trying to understand what are the receptors that sense these nutrients. At the same time, the gut has evolved a sort of a complex array of various receptor systems that sense pathogens, and today we still to understand how these connect cells are sensed and responded. So the first thing that I'll talk to you about is this increasing recognition of how dietary factors and connect cells contribute to a metabolic signaling pool that then educates gut immunity through a large panel of receptors that result in innate adaptive immunity. So broadly, when we look at the gut, there are many firewalls that restrict access of these connect cell microbes to encounter and signals in immune some of the innate adaptive systems that affect controlling responses. And Rhonda and Sheng Wei, two postdoctoral fellows in the lab, were interested in how do these microbes or connect cells interact with the mucin layer and then contribute to signaling in homeostasis. So this is a very simple approach. They ask the question, are there mucin utilizing genes, or are there enzymes and transporters that we can manually curate and then try to identify using computational approaches which are the ones that express these enzymes and transporters. So you illustrate the principle here. You look at the MUC2 peptide, it's separated by a carbohydrate chain that has terminal fatty acid or glucose that undergoes cleavage at these sites and then recognized by these transporters. So the image analysis that Sheng Wei did with help of Bert Tuckenhauer to identify these enzymes and transporters, computationally search the human microbiome one database to identify microbes that selectively express these transporter genes, whether they be enzymes or transporters. And what they observed was a clear enrichment in these class of microbes, and as we can show here, 4 of these Clostridiales species robustly utilize these single energy cells. So with this background, what Martha then asked was using a simple epithelial injury model, so the DSS model that both Andrew and Dana presented to. To ask the question, if these mice were then interbarged with these individual bugs, could you then alter the specific epithelial injury with the idea that if you don't have a mucin barrier, then you're predisposed to additional injury. So what she identified was, as expected, academic eosinophilic treatment attenuated the epithelial injury with DSS, and this has been shown by many labs. But also observed that another Clostridiales member concept, the Streptococcus fecalii, also increased weight when interbarged in the setting of this model. Examining sort of the strains that were identified in this setting, we identified that 5 strains of Streptococcus fecalii, and by background sequencing, Martha and Sheng Wei observed that there were 2 strains that expressed a phenyl acetate dehydrated gene cluster that expressed Streptococcus fecalii that initiate the sequence. So using these 3 microbes, we then did Q-TOF-MS to try to identify are there metabolites that made by these bugs that could potentially signal in this gene signaling program. So what they identified were 2 indole-containing molecules that were differentially produced by the bacteria that contain the phenyl acetate dehydrogenase cluster. So they produce, for example, indole-propionic acid and indole-lipoic acid, whereas there was a non-differential production of another propionic acid metabolite. What we also observed is that when comparing the sequences of IPA to IA was the presence of this double bond, which I'm going to get to in some of the signaling experiments. So with the availability of these 2 molecules, then Martha did 2 simple experiments. One is to ask the question, how do organoids respond to these molecules? And then how do organoids and underlying macrophages respond to these micromolecules? And notice that both IPA and IA induced the production of mucin-2, so suggesting that they signal to other cells. And also increased the production of IL-10, a cytokine that then attenuates signaling. So using a reductionist system of both organoids with and without macrophages, they show that they both have the properties of increasing the mucin-2 peptide and increasing IL-10. The phenotype of IA as well as IPA was also seen in peripheral blood cells, showing that they both attenuated the production of IL-1 beta and IL-6. If you then took these molecules and exposed them to peripheral blood cells, you can see that IA in fact and IPA to a lesser amount that increased the expression of the NRF2 genes that I listed here. And the motivation for this type of experiment was the presence of a double bond in IA compared to IPA, suggesting that this might be a better electrophilic compound. So with this data, the output is the function of the human disease relevance for this observation, and so we went back to the genomic data sets that have been generated in the lab with both HSV2 support and other funding agencies to ask what happens to these 2 clusters in patients who have inflammatory bowel disease compared to those who did not have it. As you can see here, both patients with Crohn's disease and ulcerative colitis have a reduced incidence of this phenyl acetate dehydrogenase cluster compared to the controls. So in one what I'm trying to show you is that we've used a simple computational approach to identify transporters and enzymes that are important in utilizing or metabolizing mucin. We focused on Streptococcus as an example and showed that the gene cluster here produces a restricted and metabolized indole-lipoic acid, and gone on to show that IA in fact has an influence both on the epithelial barrier as well as immune cells, and then gone on to show some of the functional indications of this observation that this cluster of these microbes in fact depicted in patients with inflammatory bowel disease. The second approach, and so what we need going forward, and I think Eric Arm will talk about this, is that we need a collection of microbes that come from humans with and without disease. We need to try to identify what do these microbes make, and then sort of embark on some of the signaling experiments that I've outlined. So the second approach that is broadly taken is to work with well-defined cohorts, and most of these experiments have been done using a cohort of about 1,000 children who were born in Finland, Russia, and Estonia, who basically carry defined sexual alleles. These children were followed for about 3 years, and the reason for focusing on this cohort is that children born in Finland have a very high incidence of type 1 diabetes, as well as various allergies to foods. Compared to children born in Russia, who had an incidence that was significantly lower, similar to what's seen in the developing world, and asked the question by sampling these children over 3 years with monthly microbiome samples, can we try to begin to understand are there microbial factors that might tell us something about this end phenotype? So tell me what that registered in the lab set about studying this, and what I'm showing you here on the screen plus, that you can clearly see that if you were born in Finland, you had a higher relative abundance of bacterial disease, whereas if you were born in Russia, had a high incidence of bacterial bacteria. So with this application, both using monthly 16S samples and monthly metagenomic samples with tables to begin to ask what are the pathways that are abundant or depleted in these two communities of homo maturing, and one of the pathways that emerged from this analysis was there was a differential abundance of linked biosynthesis and glycopolysaccharide biosynthesis, suggesting that this could be one of the pathways that could differential because of the underlying microbes that are seen in the finished state of the stage 1 in Russia.